Okay, let's get started. In a clear exemplification of the Italian saying, you give them a finger and take the whole arm, I'm chairing also this session. And first talk by Leah will be about fertilizer. So this session is about cancer resolution, and fertilizer is about growing clones. So off you go.
Do we have a clicker?
Yes.
Oh, perfect. Okay, so I'd like to start off by thanking the organizers for giving me a chance to present our work. So our method is called Phertilizer, and we're growing a clonal tree from ultra-low coverage single-cell DNA sequencing data. So as many of you are aware, cancer is an evolutionary process. So this process begins when you have a single founder cell that gains a somatic mutation. And over time, the cell divides, and its descendants may gain additional advantageous mutations, giving rise to clonal expansions or growth of a tumor. And then the result of this process is that you have a heterogeneous tumor made up of a mixture of distinct clones. So one high-resolution way that we can study this evolutionary process is with single-cell DNA sequencing. So with that, we can identify the clones and quantify the intertumor heterogeneity, as well as reconstruct its evolutionary history in the form of a phylogeny or a clonal tree. So let's say you want to study the evolutionary history of a tumor with single-cell DNA sequencing. So the next question that you have to decide is what type of technology you're going to use. So one approach is you can choose a medium to high coverage technology. So this would include like mission biotapestry or MDA. And because you have very high coverage, it's very good for performing high resolution of single nucleotide variants or SNV analysis. But because there's biases in the sequencing processes, you don't get uniform coverage of the genome, so it's very poor for performing copy number analysis. So you might consider these newer ultra-low coverage technologies. So this would include DLP plus or ACT. And historically, these methods are very good for performing copy number analysis because you get uniform coverage of the genome. However, the coverage is extremely sparse, so it's not very useful for performing SNV analysis. So after you've chosen your technology, then you have to choose your methods. So there's a number of existing methods to study the evolutionary history of a tumor. So as you can see on the left, we have the methods for medium to high coverage. And as you can see, most of these methods focus on SNV analysis, with the exception of a few of them trying to also incorporate CNA information. So on the other hand, for the ultra-low cover methods, the majority of these focus on copy number analysis. And the two exceptions being SBM clone and Cicida. So these are clustering methods, and they don't infer a tree. So the question that we're looking at in this work is, can we try to infer an SNV phylogeny from this type of ultra-low coverage data? So for my talk today, I'll start off by giving a formal statement of the problem and then describing our algorithm. Then I'll touch on our results for simulated and real data, and then end with some conclusions and future directions. So starting off with the problem formulation. So as I said, the main challenge that we have to overcome in working with this type of data is the extreme sparsity. So basically, you can see we'd have, as you get lower coverage, it becomes more challenging. And so a coverage of 0.01x basically means that only one out of every 100 bases would have a map read. So kind of an ad hoc approach, let me describe what's currently done in practice right now. So as I said, this data is typically good for copy number analysis. So what's done is first you create a matrix of your bin read counts. So you bin the genome into regions, and then for each cell in bin, you count the total number of map reads, and this is proportional to the copy number. So then you could take this matrix and you can project it into a low dimensional embedding. And then when you plot this, you could typically break out pretty clear copy number clusters. So if you wanted to perform a SNV ad hoc analysis, you can take these clusters and try to genotype those by pulling the cells together. And then you can use these tree clones and try to infer a tree on these inferred clones. So this is a reasonable approach, but there's one drawback, and that is it may not actually fully capture the SNV evolution. So you can actually have further SNV evolution within these copy number clones. So the key idea that we're utilizing in our work is that we want to use this strong copy number signal to help guide tree inference by incorporating this bin read count embedding. But we want to do it in a slightly more flexible way than fixing the copy number clusters ahead of time. And so we formulated the clonal tree inference problem. So the input to our problem is a set of variant and total reads for N cells and MSNVs. And in addition, we're going to take in the bin read count embedding that I just described. And our goal is that we want to find a clonal tree with detectable clonal genotypes and a cell clustering that maximizes our posterior probability. So for this, what do I mean by detectable clonal genotypes? So here basically we want to make sure, because the data is so sparse, that we're not overfitting. So we want to make sure that we have enough data in support of our inferred clones, and I'll go into a little bit more detail about what this means later on. So now moving on to the description of our method. So our method is called Phertilizer, and it's a heuristic algorithm to solve the CTI problem. And it proceeds in 3 phases. So you have the seeding phase. So in this phase, we basically initialize a candidate set of trees that contains only a single tree that has one node that has all of the cells in S and Vs. And in the growing phase, what we want to do is enumerate a candidate set by taking the current trees in the candidate set and performing elementary tree operations on the leaf nodes of these tree. And then the last phase, we're going to rank and prioritize our tree according to a posterior probability. Okay, so what exactly do I mean when I'm talking about growing a clonal tree? So our viewpoint basically is that every clonal tree results from a series of elementary tree operations. And so each elementary tree operation will take in a clonal tree and a leaf node of this tree and basically extends or grows the clonal tree. And in our work, we defined 2 types of operations, a linear and a branching operation. So let me describe how we would get this. Is there a laser pointer on this? Oh, here we go. This tree up here in the corner. So as I mentioned, we start off with our initial tree that has one node. And basically, we can take that as a leaf node and extend it using our linear operation. So in this, it's going to be an extension where we have a 2-node tree. And because it's a linear evolution, we would still see evidence of the ancestral cells. So the way to think about it is that the clonal genotypes are going to be fixed in this subtree. And then we can partition the cells between these 2 nodes and partition the S and Vs between the edges in this tree extension. Then we can further grow this tree using our branching operation. So again, we take in the current tree and the leaf node, and we can extend this under a model of branching evolution, where the clonal genotypes would be fixed. And now, instead of partitioning the cells between the 3 nodes, because these clones have diverged from a common ancestor, we're going to partition them between the 2 clones A and B, and then partition the S and Vs among the 3 edges in the extended tree. And then we can keep repeating this process to grow our tree even further by performing operations on clones A and B. So the question is, when are we basically going to stop this process? So that gets into the issue of detectability that I mentioned earlier. So we can think of a clone as basically a subset of cells in S and Vs. And then we can go into our data matrix. And for each cell in S and V, we could basically mark the places where we observe at least one variant read. And then we basically sum up these rows and columns, and we call these the cell or S and V marginals. And we define, say, a clone is detectable if both the median S and V and cell marginals are greater than a user-specified number t. So in our example here, say if t would be equal to 5, we would say that this, the inferred clone A is not detectable, and then we basically undo that operation. So you could potentially try a linear operation and see if that's a better fit to the data, and then additionally perform a detectability check on that. So now let me describe how we actually perform these elementary tree operations. So I said you can think about it as partitioning cells and S and Vs. So we're going to use a coordinate ascent approach, and we're alternately going to fix one of these partitions and then solve for the other. So starting with a cell partition, so this is where we're going to bring in the original key idea. We're incorporating the CNA signal via this read count embedding. And we utilize, to do the cell partitioning, we use the normalized cut algorithm, which requires basically a cell-to-cell similarity score. And so we derive this using mixing information from the S and V and CNA features in order to kind of balance the trade-off between these 2 signals. For the S and V partition, basically we're going to, once the cell partition is fixed and we have our clonal genotypes fixed, we're basically going to try to assign each S and V to an edge in the extended subtree and find the one that maximizes the posterior probability. And then finally, in our last phase, the ranking phase, we have a general model that describes our observed sequencing data. And then we want to, for every candidate clonal genotype, cell clustering, and clonal tree, we want to compute or approximate the posterior probability, and then rank and then prioritize these trees. Okay, so now moving on to the results. So I'll start off with simulated data. We wanted to verify how well our method performed when we had a known ground truth tree. So we varied the number of cells, SNVs, sequencing coverage, and clones. In addition, we compared against some other existing methods, including the kind of ad hoc baseline method that I described earlier, and SBM clone, which does joint clustering of cells and SNVs using a stochastic block model. In addition, we paired both of these methods with an existing tree inference method of site to more directly compare against Phertilizer. In terms of metrics, we looked at tree reconstruction metrics, including ancestral, incomparable, and clustered pair recall. So for these relationships, we looked at the pairs of mutations in the inferred tree and calculated what proportion occurred in the ground truth tree. In addition, we looked at an accuracy score, which is the weighted average of these recall scores. And so an accuracy score of 1 for both pairs of cells and SNVs implies that the inferred tree directly matches the ground truth tree. In addition, we looked at SNV genotype similarity, which would be the average Hamming distance between the simulated and inferred cell genotypes. Okay, so looking at our simulation results, the plot here is shown for a coverage of 0.5x. And in the interest of time, I'm not going to go into too many details on the results, so I'll just highlight our accuracy score here. And you can see that we outperform the two existing methods with the highest overall accuracy. And in addition, we see similar trends for genotype similarity, where we maintain a value above 0.95 for different levels of SNVs. So we did see a slightly drop in performance for a lower coverage of 0.01x. However, Phertilizer was still the top performing method. And our performance increased further when we increased the coverage for 0.1x. Okay, so moving on to real data. We looked at a data set of ovarian cancer cell lines. So this was sequenced with DLP+, 890 cells, 14,000 SNVs, and a coverage of 0.25x. So you can see, kind of using the ad hoc method or the baseline method that I described, you can see what these clusters look like in the embedding space. So they originally inferred 9 copy number clones and then inferred a tree on these clones. And here's the tree that we inferred with Phertilizer. So overall, there's a lot of similarities between these two trees. Both of them had 3 major clades, which closely aligned. However, there was some differences between the trees. Particular with 8 and 9, R8 and 9, and Laxon et al. Clones C and D. So let's look a little bit closer at this result here. So if we look at these in the embedding space, we can see that there's, it's not quite exactly clear. Like for A and B here, you can see they're kind of different in the embedding space, but it's not quite clear that these are 2 separate clones. So in their case, they use a density-based clustering method, which doesn't pre-specify the number of clones, so this could be the result, potentially the result of overfitting. So in our case, you see that the cells are kind of scattered throughout the cluster here. So that would be indicative of the case that I described earlier, where there's further SNV evolution within a copy number clone. So in order to kind of look at this further, we developed a metric when you don't actually have a ground truth tree, but you want to test the goodness of fit of your inferred tree. So we call this metric the cell mutational burden. And cell mutation burden works on a cell and a set of SNVs. So in this case, we're going to look at the set of SNVs introduced on each edge. And that will basically determine a clade of the tree. And it partitions the cells into 2 groups, those cells that are within the clade and those cells outside of the clade. So this basically, I'll just give it a quick example of how this metric works. So you can see in our data matrix here, we would have these 2 cells. And for each of these, we would basically mark each of these positions for the set of SNVs that we have, where we have at least 1 reference read. And then we would basically mark in orange the ones where we have at least 1 variant read. And we would just take the proportion of green X's for a particular row. So in this case, for the cells that basically are within the clade, where we would expect to have evidence of these SNVs, we would expect the CMB to be high. So in this example, it would be 0.5. For the cells outside the clade, we don't expect those SNVs to be present, and so we would have a low CMB. And also to kind of explore this metric further, we first evaluated on simulated data to kind of get a better understanding of kind of how it would work. So you can see when we calculate this on the ground truth tree, we see a good separation between the CMB metrics for cells within the clade and cells outside the clade. And we expect for the cells outside the clade to have a median of close to 0. And then we also started to experiment with, okay, if we introduce errors in terms of misclustering cells or mislabeling SNVs on the tree, what is this metric going to look like? So we see an increase in variability in terms of the cells within the clade, and then we see the median for the cells outside the clade start moving away from 0. So now we can use this metric to kind of get better insight into the real data. So again, going back to the comparison between our clade 8 and 9 and the clusters C and D. So you can see that there was very high variability for their clusters C and D, whereas Phertilizer had good separation while still maintaining a median of 0 for cells that were outside of the clade, suggesting perhaps a better fit for the data. In addition, we looked at differences between G and H, where they inferred 2 clones, but Phertilizer only inferred a single clone. And again, you can see here this could potentially be the result of overfitting, and then we do see evidence of that when we use this CMB metric. Lastly, we looked at 8 triple-negative breast tumor sequence with ACT, and you can see the coverage was significantly lower than the previous data set we looked at, with an average coverage of 0.24x. However, Phertilizer was still able to infer a tree with more than 1 clone for 4 of these tumors. However, SBM clone basically inferred only a single clone for all of these. So looking at one of these tumors in particular, for TN1, Phertilizer inferred a tree with 6 clones. And then again, you can compare the embedding from the original paper, where they used a similar embedding and clustering approach to define a set of superclones with the Phertilizer cell clusters. So overall, there's good alignment between these cell clusters. However, again, Phertilizer refines some of these copy number clones further, and those are supported by our CMB metric. So in conclusion, we showed that it is possible to infer an SNV phylogeny from ultra-low coverage single-cell DNA sequencing data using our method Phertilizer. So this will now allow people to basically perform copy number analysis and SNV analysis from the same set of cells. So in terms of future directions, we'd like to look at incorporating CNA events directly into the clonal tree, as well as extending Phertilizer to make use of other evolutionary models and omics modalities. So I'd like to acknowledge my co-authors, in particular my co-first author, Chuanyi Zhang, our funders, and our other lab members. Thank you.
Thanks a lot. We got time for a couple of questions. I have a question. All right. This sounds like a very urgent one.
No, because I thought you cannot see me, that's why I...
And that's true.
Okay, so the question I'm having is, how do you decide on the number of copy number clusters when you do clustering based on copy numbers, and how do you go from this clustering to tree inference?
Yeah, exactly. So that's the kind of the advantage of our method is we don't limit it to, we don't specify the number of clusters. So we basically just grow the tree until we don't basically have enough data to basically refine it any further. So we do this as we're kind of building the tree using these elementary tree operations, and we'll basically just take the set of cells in the leaf node and try to partition those further, and then look at the goodness of fit of that. So we don't actually have to decide the copy number clusters ahead of time. So we're just
using Chisel for inferring copy numbers of individual cells, and then if you know copy numbers, you can cluster cells.
Yes,
easily.
yes,
Have
yes.
you tried that?
So that's kind of the ad hoc method that I described. So you can, that's kind of a current approach that's used in practice. So basically you break out the copy number clusters, and then you can genotype those and then infer a tree. But you may be able to refine those clusters further. And so our method is kind of using that still using the embedding information to take that approach instead of fixing them ahead of time.
There was a question, Carl, perhaps?
Actually, I have two if I can.
Okay.
So
Allow.
first one is you have a posterior posterior distribution in the end.
Yeah.
Do you use any kind of prior, especially on copy number states?
Yeah, so we don't have a particular prior on the copy number state, so we're not actually inferring those. We're using the recount embedding a bit as a proxy for that. And so we don't really have any prior on the trees per se, so we kind of construct those through our growing process. So we're basically not really like sampling from our generative model, we're kind of using a heuristic algorithm and just using our generative model as a way to calculate the posterior probability.
Yeah, I would have one.
Oh, sorry.
So I didn't understand exactly if you have to do you have to enumerate each possible clonal tree and then like do a comparison, or do you have any kind of like method to decide in which direction to go with the elementary operations?
Yeah, that's a great question. So currently we're enumerating them. So we're basically trying all possible combinations of linear and branching on each one, and then growing our candidate set. So as I said, one thing is like working with this data is very sparse, so you're actually not going to, the trees aren't going to get that large, so it makes it still tractable to do that. But there, like some things that we've been thinking about, okay, is there a way to kind of like prune this process so you don't have to look at trees that are not promising? So.
It was a beautiful talk.
Oh, thank you.
Yeah, I think slightly related to the earlier question, but is there utility if you know part of the tree, say you've got very good quality sequencing, it tells you some structure of the tree, can you use that as a kind of prior rather than having to do everything de novo? Does that help?
Yeah, I think so. We haven't explored that, but I think that would be a good suggestion. You could just basically try to maybe like refine or validate certain parts of the substrate or try to extend them further. So I think that would definitely be possible.
There was a question.
Yeah, back here at that.
Question here.
Okay, I want to ask whether the high sparsity induces the tree to be not balanced and then affect the performance.
I don't know per se if the sparsity would make the tree unbalanced. I think that would be kind of more a function of the evolutionary process.
Maybe all like
Yeah, it makes it harder to depict. Yeah, it might make it a little bit harder to infer one that's, yeah, potentially. I'm not too sure about that. We haven't looked at that. But.
Then we have a final question here.
Yeah, it's a technical question. So why are you calling this method fertilizer? What is the significance?
Oh, so fertilizer, like with an F, is when you like try to help your lawn grow or your plants grow. So we're trying to help our clonal trees grow. But yeah. So if you put a pH, it's for phylogeny. So.
Now that everything is clear, let's thank.
Okay.